Want to learn about the structure of DNA? Then you're at the right place. Many students forget that DNA stands for deoxyribonucleic acid. DNA is a molecule made up of two strands that are twisted around each other in an alpha helix shape. Now, each of these strands has a 3' and a 5' end. They run in opposite directions and therefore are said to be antiparallel. Each strand is made up of nucleotides which contain three important components: a phosphate group, the nitrogenous base, and a deoxyribose sugar. Now, let's look at the nucleotide closely. It's important to know how to label these three important components. So first up is the nitrogenous base right here. In the middle, we have the deoxyribose sugar. It's also called pentose sugar. And then on the top left, we have the phosphate group. You might be asked to state the carbon number to which the components are attached to. So let's go ahead and look at that. At carbon 1, the nitrogenous base is attached to the deoxyribose sugar. At carbon 3, the next nucleotide is attached to this nucleotide. And at carbon 5, the phosphate group is attached to the sugar. Now let's look at how these two strands are even held together. This is possible due to the complementary base pairing, which refers to the specific hydrogen bonding between the nitrogenous bases in DNA. The nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine. Adenine and guanine are both called purines, whereas cytosine and thymine are both called pyrimidines. The adenine pairs with thymine through 2 hydrogen bonds, whereas the guanine pairs with cytosine through 3 H bonds. Remember that purines always pair with pyrimidines. This is very important to remember. You might be wondering about the difference between purines and pyrimidines. Well, as you can see on the diagram, purines have 2 rings, whereas pyrimidines only have 1. That's it for this video. Thank you for watching.